Hello, and a very warm welcome to today's Nucleic Acid Insights webinar, titled Enhancing mRNA Production with Advanced Analytics with WACKER Biotech, which is the second session in our virtual event, Biopharmaceutical Analytics Unveiled, Expert Insights and Customer Success Stories. I'm Charlotte Barker, an editor at BioInsights, and joining me today are Blaž Bakala and Michael Daume, who will discuss how mRNA production faces challenges in separating complex IVT mixtures and how WACKER Biotech uses the PATfix platform to streamline process development, enabling precise quality control and impurity identification. After the presentation, we'll have a live Q&A session, so feel free to pose your questions to our speakers using the Ask a Question box at the bottom of your screen, and we'll try to get to them during the session. Now I'd like to introduce our presenters. Blaž Bakala has a PhD in nucleic acid biophysics and is now the PATfix product manager at Sartorius BIA Separations. Michael Daume, with a PhD in small RNA biology, is now a research group leader at WACKER, focusing on downstream processing development for plasmid DNA and RNA purification, leveraging his expertise in chromatography and mRNA process development. So without further ado, I'm going to hand over to Blaž now to get us started.
Thank you, Charlotte, and welcome back, everybody. So we've just discussed in the previous session how to make pDNA, and we ended up with linear DNA. Now let's explore a use case where this linear DNA comes into play, which is in the production of mRNA. And looking at the mRNA process, we would start on the left with our linear DNA, which we would use in the IVT reaction. We would then do purification, final formulation, filtration, and have a finished drug substance. We offer here many different approaches when it comes to the capture and polishing of the mRNA itself, but today's focus is going to be more on how analytics can support this process, both IVT production and then downstream purification. So the IVT reaction, the very interesting part of mRNA production. And it's important to stress here that this is a reaction and it happens very quickly. Usually it takes 2 to 3 hours for batch processing. Is very high yielding. Just recently, our group published a paper where they were able to reach about 25 milligrams per milliliter of mRNA produced. The IVT reaction itself has multiple components, and these components are very important when it comes to understanding the kinetics of mRNA production. So we would, of course, have our DNA template, the enzymes, the NTPs, the capping reagents, the buffers, and other additions that can be added. Now, for the capping reagent, of course, there's two approaches here. There's co-translational capping and there's post-translational capping, transcriptional capping. And for the purification, having the ability to monitor all of these components, both NTPs that are the building blocks, as well as the capping reagent and the mRNA that's produced, allows you to optimize the IVT reaction to a very high yield. But even after the IVT is optimized and you have the mRNA, raw material impurities from the plasmid and DNA, so process-related impurities like the plasmid DNA, the enzymes, and other reagents like nucleotides are present, and monitoring the removal of those is important. So the way we achieve this is we developed the analytical platform specifically for this, where the most utilized and interesting method is, of course, for IVT reaction monitoring, where we can separate all of these critical components like the capping reagent, the nucleotides, the template, and the produced mRNA, which then allows us to take a time course as the IVT is going and monitor how the mRNA is being produced. Beside that, the platform also offers quantification of pure polyadenylated mRNA using affinity analytical column with an oligo-DT ligand, as well as controlling mRNA integrity using the SDVB column, which shows patterns if mRNA degradation occurs. So this is how a time course of an IVT would look like, where you have the possibility to sample every 15 to 30 minutes usually. And as soon as you sample, you can put sample on the PATfix and get the resulting NTP and mRNA concentrations, which then allows you to basically very easily track how the reaction is performing. Not only just the mRNA production, but also consumption of nucleotides. The consumption of nucleotides here is especially interesting because if we look at the bottom left, we can see that the mRNA, the IVT reaction has basically stopped at about 120-minute mark because we ran out of the red nucleotide, which in this case was ATP. So if you were to act on this knowledge and wanted to go even higher in your yield, you can add additional ATP to the starting IVT, which will allow you to achieve higher yields in a subsequent IVT reaction. And using this approach allows you to basically very quickly iterate on your IVT optimization process to reach the desired results when it comes to purity as well as yield. Whereas traditionally, you would collect these samples in a limited fashion and send them off to another analytical laboratory for analysis. What we offer is basically at-line monitoring, so you can have the results in the same day and act on them the very next day, which allows you to then, for instance, even utilize tools like DOE to increase the IVT production, the mRNA production via IVT even further. So the PATfix mRNA platform is designed to basically robustly analyze the mRNA production and purification samples. It doesn't come just with the hardware. There's also guides included in there on how to prepare every process sample to basically achieve good results. It's very fast. The methods included in the platform are usually between 3 to 15 minutes long. And it doesn't cost much compared to other analytics to run. And by leveraging this platform for which we have developed analytical methods, you don't need to go out and search for the right hardware and the columns, develop your internal standard and optimize the analytical method for yourself. You can get these results pretty quickly at line. Now, to tell us a little bit more about this, I'm going to hand off to my colleague Tina, who's going to show us how to use the PATfix mRNA platform practically as a user.
Hi, I'm Tina Vodopivec-Seravalli, and I'm part of the pDNA and mRNA Process Development Group. In this video, I will present you the mRNA analytics with our PATfix platform. The recent pandemic significantly accelerated the development of new messenger ribonucleic acid vaccines and other therapeutics. This rapid advancement was driven by the urgent need of effective treatments and preventive measures. Consequently, the in vitro transcription reaction, which produces mRNA, has become the focus of intense research and development. The primary goals are to increase mRNA production while simultaneously reducing its high cost and minimizing unwanted byproducts. The IVT reaction is relatively complex, involving many different components. The most important among them are capping reagent, nucleotides, pDNA template from which the mRNA will be transcribed, various enzymes, with mRNA polymerase being the most crucial one, and magnesium ions, which we have found to be very important in the IVT reaction. Under the right conditions, mRNA production continues until the nucleotides are consumed. Optimization of IVT depends mainly on understanding the impact of each reagent on the kinetics of mRNA production and the consumption rate of each building block. Low throughput and endpoint-only analytics do not provide the insight needed and therefore can significantly slow down the process optimization. Agarose gel electrophoresis, for example, is a semi-quantitative method that provides limited insight into your sample components. It takes approximately 2 to 3 hours to visualize and evaluate a limited number of samples. Now let's say we are running an IVT reaction, and at the end, we want to determine if we were able to produce any mRNA. After 4 hours of IVT incubation, we inactivate the sample and proceed to perform an agarose gel electrophoresis analysis. At the end of the working day, when the results are ready, we see that our IVT reaction didn't produce any mRNA at all. We evaluate our results and decide to try a different IVT protocol the next day. But even when we do manage to produce and visualize some mRNA, several questions still remain. How do we know if and when all NTPs were consumed? How much of the NTPs remained in the reaction? How can we precisely quantify produced mRNA? The need for an at-line method for IVT monitoring and mRNA quantification was higher than ever. In response, we developed a chromatographic method using our CMAC'-S column. Today, this method is one of 3 validated methods for mRNA production and purification monitoring available in our PATfix mRNA platform. Let's dive into the world of ''S analytics and IVT monitoring. First, let's talk about the column itself. Like our other analytical columns, CMAC'-S is packed in a stainless steel casing, allowing it to withstand higher pressures. ''S is a multimodal chromatography ligand that combines element of hydrogen bonding with anion exchange chromatography. The method we developed and validated has a 4.5-minute readout and is based on the separation of nucleotides in an ascending salt gradient and the elution of mRNA under high conductivity and pH conditions. With our method, we are able to separate capping reagent, nucleotides, DNA linear template, and of course different modalities of RNA. CTP and UTP do coelute. Most of the time, this does not cause a problem, as ATP or GTP are usually the limiting nucleotide in an mRNA sequence. For when it does matter, we offer a mathematical formula that helps track the individual concentration of UTP and CTP using their different extinction factors and absorbance ratio at UV260 and UV280. When performing an optimization of any kind, it is important to have an inside look at the reaction itself. For IVT reaction optimization, the most important part is understanding the kinetics of the reaction. Key factors include how quickly are the nucleotides being depleted, the rate at which mRNA is being produced, and transcription factor, which shows the mRNA concentration relative to the linear DNA template concentration used in the IVT reaction. PATfix analytics using CMAC'-S column can answer all of these questions. To prepare for IVT monitoring analysis with PATfix, first we need to connect the buffers and the column to the system. After flushing the system with buffers, we run at least 3 blanks to ensure that the baseline is stable. Next, we inject the system suitability test sample to check the column conditions. If the separation of all components is within expected parameters, we can proceed to the injection of the calibration curve made from our provided mFix4 standard, and after that, our samples. When optimizing an IVT protocol, we sample the reaction over time, usually every 15 to 30 minutes, depending on the protocol we are using and the results we are expecting. We take 2 microliters of the IVT reaction and inactivate it with 2 microliters of EDTA. This binds the magnesium ions, making them unavailable to the RNA polymerase and effectively stopping the reaction. Before PATfix analysis, the sample must be additionally diluted 100 to 400 times with mobile phase A. The dilution is determined based on the predicted end mRNA concentration in the IVT reaction. For precise calculation, you can use the formula from our paper, or, to simplify, the end mRNA concentration in milligram per milliliter corresponds to the starting concentration in millimoles of the most frequent NTP in the mRNA sequence. For example, if we add 5 millimoles of ATP to the IVT, we can expect 5 milligrams per milliliter of mRNA if the ATP is the limiting nucleotide. After the samples are analyzed, we can easily visualize the chromatograms in the PATfix software. When analyzing the data, first we create the calibration curve and apply the calibration to our analysis. After we integrate a sample, its concentration is displayed. After evaluating all results, we can determine the optimal conditions for the IVT reaction depending on our specific goals, whether we aim to produce mRNA more quickly or reduce the cost of the IVT process. The data will guide our optimization efforts. In this experiment, for example, we aimed to determine the importance of the concentration of magnesium ions in an IVT reaction. We designed an experiment where each IVT had a different magnesium chloride concentration. If we only looked at the final points, we might falsely conclude that we only had 2 different outcomes. 2 reactions performed poorly, while all the rest had the same final mRNA concentration. However, by examining the reaction kinetics, we can clearly see that some reactions produced mRNA faster than the others and were completed in under 1 hour. This allows us to choose whether we want the reaction to finish faster, using higher reagent concentrations, or to lower the cost of the IVT by allowing the reaction to run for a longer time. Choosing which one to optimize often depends on our available resources and overall stability of the mRNA at higher temperatures. If needed, we can also monitor the course of the IVT reaction in real time. This is especially important when planning to fit additional nucleotides into the reaction. By monitoring the remaining nucleotide level, we can decide how much of each nucleotide to add to prolong mRNA production and achieve better yields from the IVT reaction. Additionally, when trying new protocols, buffers, or reagents, we can act quickly if we notice that the reaction is not producing any mRNA. By monitoring the reaction in real time, we can attempt to correct the IVT by adding specific reagents, thereby gaining a better understanding of the process and potentially salvaging the IVT. For example, in this reaction, we were testing a new commercially bought buffer. The reaction was not proceeding as expected. After 3 hours of incubation, with little to no mRNA production, we decided to add some magnesium to the IVT. Already at the next time point, we observed mRNA production, confirming that our hypothesis was correct and the reaction just needed more magnesium. In mRNA production, the most expensive step is the IVT reaction due to the high cost of capping reagents and enzymes. Optimizing this step to achieve higher yields is crucial for reducing overall costs and minimizing process impurities. The PATfix mRNA platform with IVT monitoring method allows you to separate all main IVT components and track them over time to determine the reaction kinetics. You can also use the same method to monitor optimization of mRNA purification steps, where you can identify potential residual impurities and calculate the recovery of each step. To learn more about the PATfix mRNA platform and other methods for mRNA analysis and quality control, visit our webpage where you can try our PATfix demo software or contact our sales team with specific questions.
Wonderful, thank you. Great presentations. Now we're going to pass over to Michael to start his presentation.
All right. Hello, everyone. Okay, first of all, thank you for the kind introduction. I highly appreciate your invitation to this webinar and giving me the possibility to present WACKER Biotech to this audience and show our analytical and manufacturing capabilities for mRNA production. However, I have to be honest, so when Blush asked me around 2 months ago if I want to be part of this webinar, I was a bit hesitant of accepting the invite. The problem was that we just recently purchased a PATfix device, and I was just too worried if we would have enough data to show. However, my worries were unfounded because the device is really straightforward. So it comes with well-developed methods and foolproof SOPs that really minimize your developmental activities. So you can basically start right off measuring your samples. All right, let's start into the talk. In the first part of my talk, I would like to present my company. So many of you may not associate WACKER with life science industry and may rather know WACKER as one of Germany's biggest chemical companies. We have over 16,000 employees, and we are large producers of polymers, polysilicon, and silicones. But we also do have a large biosolutions division and produce bioingredients like cyclodextrins or cysteine, for example, used in baby food. Part of this biosolutions division is WACKER Biotech. This is a 20-year-old contract manufacturing organization with own developmental activities and EMA and FDA approved manufacturing sites. The biopharmaceutical products we produce for our customers, they range from live microbial products to classical vaccines to therapeutic proteins and to products used in advanced therapies, meaning plasmid DNA, mRNA, and lipid nanoparticles. WACKER Biotech globally operates 5 GMP facilities and 1 R&D center. 3 are based in Germany, 1 in the Netherlands, and 1 in US. Plasmid DNA is produced in Halle and in San Diego. RNA and lipid nanoparticles are produced in San Diego, Amsterdam. And in our newest site, also placed at Halle, this is our new mRNA Competence Center, and it opened just in June 2024. And we are really proud to announce that we were awarded the contract by the German government to ensure the production of 80 million doses of vaccines, RNA vaccines, in the event of another pandemic in this site. And in Munich, where I am based, is the corporate's R&D center. Here, 7 different research groups are working on the development of novel technologies for the production of plasmid DNA, mRNA, and lipid nanoparticles. My group, for example, works on the development of downstream processes for both plasmid DNA and mRNA. Okay, so WACKER Biotech is a real end-to-end mRNA CDMO expert. That means we start on gene level. So we assist our customers in mRNA design or also already on template level. We produce the pDNA by microbial fermentation, mRNA by in vitro transcription. We formulate it into lipid nanoparticles, and we have our own fill and finish line. At the R&D center in Munich, we have an own department just dedicated for nucleic acids. We are always staying aware of the IP landscape, which is quite complex actually for the mRNA field, but try to provide proprietary solutions for our customers and offer processes that are freedom to operate where possible. We have our own in-house lipid design and a cell culture lab that ensures that the LNPs we produce are in fact functional, and our customers also appreciate our speed. So we are capable of providing R&D grade material within 1 to 9 months and GMP grade material within 12 months. We also have a good GMP track record. So we produced batches with 80 grams of RNA per batch, and we are, as I already said, part of this German pandemic preparedness program. And we have a wide range of scales. So we are capable of producing pDNA in fermenters of up to 1,500 liters, mRNA up to 30 liters, and lipid nanoparticles in up to 3,000 liters. Okay, so WACKER's corporate R&D has a strong network for advanced therapeutics. We have set up many collaborations with universities like the Humboldt University in Berlin, with the Technical University in Munich. We have even set up a new institute for industrial biotechnology, where WACKER is funding a large number of PhD projects. We are part of several research clusters, for example, CNAT-M for nucleic acid therapy research, or the Saxo-Cell cluster very newly. And we have built up early collaborations with biotech companies like SRTD Biotech or Panthera Therapeutics, where I will show a case study in a few slides. And we assist these companies scientifically, for example, in the design of the RNAs or pDNA, and by providing preclinical material, and this encompasses pDNA, mRNA, and lipid nanoparticles. All right, what are the solutions WACKER Biotech has for the manufacturing of plasmid DNA? We have our own proprietary strains and plasmid, which we term PlasmidTech, and use a cutting-edge USP fermentation protocol that allows us to achieve high yields of plasmid within a fast batch process with titers that can keep up with the leading competitor in the market. Our PlasmidTech strain and the adaptation of the USP protocol already allows high purity of supercoiled plasmid DNA during fermentation, and our cutting-edge DSP process is capable to purify supercoiled pDNA to purities of above 95%. We also work on innovative technologies. For example, we recently patented a new approach for faster and higher pDNA production in E. coli. Or, for example, we have worked on a new approach for plasmid DNA linearization using a CRISPR-Cas9 approach. All right, what are the solutions we have for mRNA manufacturing? We are capable to achieve high yields during IBT reaction, usually in a process that involves post-transcriptional capping. This is also what we suggest to our customers. Sorry, somehow the numbers are a bit off here. But you also don't have to take a loss in capping efficiency for these post-transcriptional capping. So we see very similar capping efficiency. And also our RT-PCR-based method of poly-A length determination shows that the RNAs or the poly-A lengths we produce also match with the aimed size. Our DSP process is capable of reducing double-stranded RNA amounts to below 1%. All right. I don't know what happened to this presentation, but somehow the numbers are a bit off here. Okay, so in this slide, I would like to bring up a success story where I want to show how WACKER's R&D Center efficiently produced research-grade material with high purity for a customer. So we have started a collaboration with Panthera Therapeutics. Panthera is developing mRNA therapies that show tissue specificity by using a combination of 5' UTRs and special LNP formulations. for example, against the acute respiratory distress syndrome. The target mRNA expression protein here is a cartilage oligomeric matrix protein called COMP-ANG1. And we assisted Pantherna by the design of the mRNA and by providing R&D grade material with purities of above 90% and 95% capping efficiency within just 3 months. And Pantherna tested this WACKER process of pDNA templated mRNA production and post-transcriptional capping against a co-transcriptional capping process. And we're able to see that the relative COMP-ANG1 expression in vitro was higher using this WACKER process. And Pantherna was so convinced in our work that they in the end decided to go into mRNA production at our new competence center in Halle, which of course makes us really happy. All right, in the second part of the talk, I would like to put the focus more on mRNA analytics because that's what this webinar is about today. First of all, I want to say that we try to keep our analytical methods up to date with the current guidelines. So what I'm aware of currently is the third draft guideline by the US Pharmacopoeia. I think it came out in August this year. And marked in bold are methods where we now use, in addition, a PATfix device to analyze our mRNA. So what is the portfolio that we have? So for mRNA sequence confirmation, we use a next generation sequencing approach. For RNA concentration, we use UV-Vis spectroscopy or fluorescence. For mRNA integrity, we analyze our samples now with an ion-pair reverse phase HPLC chromatography using PATfix, besides capillary gel electrophoresis and agarose gel electrophoresis. mRNA purity, we additionally added a mixed mode HPLC method by a PATfix device to this list. 5' capping efficiency is analyzed by an RPLC-MS method, the 3' poly A tail length by RT-PCR-based method in combination with capillary gel electrophoresis, and residual double-stranded RNA is analyzed in an HT-RF assay that is similar to an ELISA assay, but it's based on FRED. And fragmented RNA, I will later show some data where we also now use this ion-pair reverse phase chromatography using PATfix. Residual template DNA is analyzed by qPCR and residual protein by BCA and silver staining. Expression of a target protein is usually at the customer site, but we can assist with our cell culture lab in the development of assays if this customer is still in preclinical phase. And for standard methods like endotoxin or appearance, we use standard pharmacopoeia methods. All right, now to some real-world data. So I would like to do a short excursus to plasmid DNA, and more specifically, the plasmid DNA that is to be linearized as a template for mRNA production. So here we have tested a different buffer composition or different buffer compositions for linearizing pDNA with X-bar1 enzyme. And while agarose gel electrophoresis gives you already a good impression of how the linearization status is, a quantification of linear and circular plasmid DNA is quite cumbersome and error prone. And the PATfix device provides a real remedy here. So we use the CMAC pDNA method to analyze these samples. First of all, in the uncut samples, it's nice to be capable to separate the open circular form and also the supercoiled form. But with this chromatography, you're even possible to separate the linear form from the circular form. And with this, you can easily quantify the amount of linearization within the reactions. So here, for example, for buffer A and number 4, only at this point you have 100% of linear plasmid DNA. And this method is also quite fast, so you get your first result after 10 minutes. And this is, of course, perfect for at-line analytics, where you want to say or want to know at what point is your reaction fully linearized, and you can proceed to save time and costs. Okay, let's get to the next slide. We use the CMAC Prima S HPLC method to analyze our IBT reactions. So on the left side, you see an overlay of Prima S chromatograms, where we took every 15 minutes a sample from an IBT reaction. And you can see that the NTPs are being separated on this column, also your linear plasmid DNA and your mRNA. And you can nicely see how the NTPs are decreasing over time as they are being incorporated into mRNA, and how the mRNA absorbance increases. This method is even faster than the CMAC pDNA method, which is really great to react to these IBT reaction, as it was shown in the video before. So on the right side, you see a function of mRNA amount and NTP amount against time for 2 different RNAs, Cas9 and fluc mRNA. And you can nicely see how after 105 minutes, your mRNA production comes to a halt. And the reason for this is that the ATP is being used up here. So this makes this method perfect for fed batch approaches. So if you would spike some ATP in here, as Tina had shown in the video before, you would be capable of increasing your amount of mRNA in this reaction. In comparison, when you look at the fluc mRNA, you have your NTPs available until the end of the reaction. And this leads to basically a constant increase of mRNA. And I would like to make the point here that it's really critical that you understand your IBT reaction. So each individual component in this reaction has an impact on your kinetics of the mRNA. And you really need to optimize your IBT reaction to each construct. And this is why this PATfix method here is so powerful, because you can easily adapt. All right. We also use the CMAC Prima S method to monitor our DSP steps. In this case, we performed an oligo-DT chromatography run to purify our mRNA. You see your IBT load. So this is the flow-through fraction. This is the wash fraction. This is your eluted mRNA and water. And agarose gel electrophorezes and capillary gel electrophorezes, they give you a good impression of what the integrity and the quality of the RNA looks like. However, you don't get really much information about the purity of the RNA, especially in terms of small components like NTPs. And these you can see in the Prima S chromatogram. So here, for example, for the IBT load, you have your still remaining NTPs and your mRNA from the IBT reaction. And then in the wash fraction, you see how the NTPs are washed away. A little bit of mRNA is lost also, however, not much. And then in your elution fraction, you have your pure mRNA, which is free of any NTP contaminants. We didn't use a capping reagent here, so if you would have added here, you would also see it in the chromatogram. All right. Then last but not least, we also use SDVB chromatography to analyze our mRNAs. SDVB is an ion pair reverse phase chromatography. And first of all, I would like to show that you're capable of separating your RNA by size with this method. So here for the high range RNA ladder, you can nicely see from 200 to 6,000 nucleotides how the RNA is being separated. Also, our 4 mRNAs we tested, EGFP, fluc, mFix, and Cas9, are separated by size. However, you are not capable of doing exact size determination using this method. And that's why I only show data here for capillary gel lactophoresis, where we see quite well matching results between the theoretical size of the RNA, at least for Fluke and EGFP mRNA. For Cas9, we do see some offset, which we often observe for RNAs that are larger. But we used this method for integrity analysis, and there was a nice comparative study by BS Separations last year in Cell and Gene Therapy Insights, where they compared integrity analysis using SDVB versus CGE. And we used their technique of peak integration, and could see that we had very comparable results between CGE and SDVB analytics. Better for Fluke and EGFP mRNA. For Cas9, we had around 8% difference, which is probably still acceptable. I saw that, so I read this paper again, and I found that they also observed up to 8% difference for their RNAs. For them, it was more or less like an outlier. To say that this is these are the first mRNAs we are analyzing on this method, and we just have to generate more data to see if we observe this more often, or if this is just an outlier here. All right, I would like to sum up this talk. I presented you WACKER Biotech, who is a CDMO expert for advanced therapeutics with over 20 years of experience. We use a cutting-edge analytical portfolio to determine the quality of our nucleic acids. And I showed you how we use PATfix to monitor and optimize our enzymatic reactions like pDNA template linearization and mRNA production, and verify the purification status during these P steps, and determine the integrity of our mRNAs. And at the end, I would like to encourage you that if you're interested in a scientific collaboration project, or need preclinical material, or if you're in search of a very talented CDMO, please just write me. We would be really happy in helping you creating tomorrow's solutions. And with this, I'm at the end of my talk. Thank you very much for your attention. I would like to thank Pantherna for giving us the possibility to show their case study, and I'm curious about your questions. Thanks a lot.
Brilliant. Thank you very much, Michael. Great presentation. So before we're going to start our Q&A, we'd just like to ask the audience a quick question, which should be popping up on your screen about now. I'll just give you a few moments to answer that, and then we'll get to the Q&A. Brilliant. Thank you very much for your input there on the survey. Now we're going to start the Q&A. So I'd like to invite Blaj and Michael to join me on camera. Brilliant. So yeah, I hope you're ready to answer some questions. So first question I've got here, why is mRNA size determination not possible on the SDVB column when RNA can be separated by size? Michael.
Let me quickly share these slides again. I can switch to the slide. So the thing is that the SDVB chromatography is it's an ion pair reverse phase chromatography, so it is denaturing. However, your mRNA is also separated by hydrophobicity, and that means so if you have two different RNAs of the same length but different nucleobase content, you basically have a different retention time. So more hydrophobic RNA will elute at a later time point. And that's why you basically cannot use this method. So for example, for the Cas9, which is around 4,500 nucleotides, it comes down here behind the 6,000 nucleotide peak of the high range RNA ladder. Yeah.
Great, yeah, that makes sense. Another question for you, Michael. Why is the mRNA present in two peaks?
Okay, let me switch to this slide. So, Blaž, you may need to correct me, but I hope I'm right. So the PRIMA-S technology, it's a mixed mode chromatography, and it uses anion exchange chromatography in combination with hydrogen bonding. And your mRNA elutes based on a change in conductivity and also in pH, and your gradient of the elution buffer. So your change of conductivity and pH doesn't happen at the same time, and that's why you have this offset. And you probably have two different conformations of mRNA which elute, at these two different time points. But I know that BS Separations is already developing a new method where they're capable of, in the end, achieving one single peak for the mRNA. However, it's not rolled out yet to the PATfix software. So I'm really curious about this one in future.
Yeah, that's exactly correct, Michael. The two peaks that we observe, we linked them probably to different mRNA conformations being present on the column that gives them slightly different behavior. However, we have managed to recently modify the method so that the mRNA itself elutes as a single peak, and we'll be rolling that out as soon as we extensively validated and tested the method to the PATfix platform.
Brilliant, thank you both. Okay, question for you, Blaž, now. How did you decide the amount of each NTP? Was it depending on the length of mRNA? That's the question.
Right, so the initial concentration of the NTPs is usually linked to the sequence composition of your target mRNA to be produced, of course. And the higher you go with the NTPs, the higher you can push the yield of the mRNA itself.
Perfect, thank you. And why does CTP and UTP elute in one peak? Is it possible to separate them? Michael, would you want to take that one?
Yeah, I think so. It's currently not yet possible to separate them. I think also BS Separations again is developing a new method where they actually can separate it. But so as Tina showed in this video before, you have this different absorbance between UTP and CTP, and you can use a standard with different ratios of these two NTPs to get a rough estimate of how much of the different NTPs you have in your sample. So that helps a little bit.
Yeah, that's exactly correct. We've also listened to users, and we know that that's an area of interest. And again, there is a method in development to specifically address that, and have those two separated as well.
Great. So watch this space. Okay, this is for Michael. What is this laddery pattern that you can observe for your mRNA in the agarose gel?
Yeah, let me share my screen. So I guess you mean that one. Yeah, so I'm not 100% sure. It's no plasmid DNA. So here we use the PCR template for mRNA production. It might be some secondary confirmation of this mRNA. It is present in the IVT load and wash fraction where we have some salt in there. So it might be an artifact actually that comes from the addition of the salt. We don't have it in the elution fraction. So yeah, I cannot really tell where it comes from, but of course I'm happy if I lose it in the end and we have our single band in the elution fraction.
It's a bit of a mystery, that one. If NTPs are sufficient in IVT reaction, what problems do occur? Blaž?
Well, if they're sufficient, usually there is no problems. However, what can happen, and this is something that Tina and Michael have shown it very nicely, is that there's each component in there, from magnesium to pyrophosphatase to the individual nucleotides, affect pDNA, temperature, pH. We can go on with the factors. Affects the kinetics of the reaction, and can even stall the reaction as well. So as long as nothing's going wrong, everything's okay. But as soon as something goes wrong, you want to figure out why. Having this analytical insight is quite good to help you basically resolve that very quickly.
Yeah, definitely makes sense. Michael, why do you see such a size difference in Cas9 mRNA during capillary gel electrophoresis?
I think we probably have to optimize this method a little bit better for a larger RNA. So we always observe this for larger RNAs. Problem is that the retention time difference between large RNAs gets smaller and smaller in the CGE, and maybe we have to play around a little bit with the gel concentration also to have a better separation of these large RNAs. So I think there's still a little bit of optimization necessary for large RNAs with this method.
Perfect. And what does the DF curve in your SDVB chromatograms represent? What do you use it for?
Yeah, let me show this slide again. So you talk about this curve here. So this is basically the first derivative of the tangent. So it describes the maximum slope of the absorbance signal. And this is a tool integrated into the PATfix software, how to determine where you basically have to draw the line between the fronting and the main peak. So the fronting is basically fragmented RNA, main peak is your full-size RNA. And BS separation is nicely shown in their paper that when they do a fraction collection of these two peak areas and load it onto capillary gel electrophoresis, that they see for this fronting area really only fragmented RNA, and for the main peak area, only the full-size RNA. And I really like it because it is less subjective instead of like just drawing a border. Yeah, so this is what it represents.
Brilliant. We've got time for one more then. Can you separate N1 methyl pseudouridine modified mRNA from non-modified? Blaj, do you want to take that one?
Oh, that's an interesting one. I'm not sure we actually looked into that one yet. However, when it comes to pure nucleotides, again, this is one of the methods that we're also working on, because through feedback from customers, we noticed that there's also a desire to separate N1 methyl pseudouridine from the rest of the nucleotides. So that will be available in the future. However, when it comes to modified RNA versus non-modified RNA, that's an interesting question, and we'll reach out after the talk and maybe discuss this.
Yeah, sounds like an interesting one to explore further. Well, wonderful. Well, thank you very much, Michael and Blaj, for answering those questions. That is all we've got time for today. Any questions that we didn't get to, or reply by email. This webinar will be available on demand tomorrow, so look out for an email from us with the link. We'd also like to invite you to take a short survey to provide us with feedback on today's webinar. The link to that can be found in the Session Resources tab on the left hand of the screen, or it will also be in the on-demand email. So all that's left then is to thank our speakers once more for a great presentation, and thank you to the audience for watching. We hope you join us in the next session, starting in a few minutes, which will cover advancing LNP characterization for drug delivery. You can find that by returning to the lobby.